Assalamu alaikum wa rahmatullah wa barakatuh. Distinguished guests, esteemed colleagues, ladies and gentlemen, welcome to this afternoon's keynote lecture. I am Omar Kniou, Professor of Applied Mathematics and Computational Science, Chair of the 2018 Enrichment Program. Before introducing today's speaker, I would like to acknowledge the participation of colleagues from 3 Saudi universities, King Abdulaziz University, Al-Qura University, and the University of Jeddah, who has a delegation led by Dr. Ramzi As-Saadi, Dean of the College of Science. Let's extend a special welcome to all our guests. As you may know, events organized by the Enrichment Program are the result of collaborative efforts among all the divisions of the university, including the academic divisions, the Office of the President, Saudi Affairs, Government Affairs, Community Life, and Facilities. The 2018 Fall Enrichment Program featured a special collaboration, namely with the Red Sea Research Center. I would like to thank Professor Michael Berryman and the RSRC team for their help in both designing and executing the 2018 Fall Enrichment Program. I would like also to extend special thanks to the team of the enrichment programs. It certainly has been a privilege and a pleasure to serve with them over the past year. Let's show our appreciation for the enrichment team and all our partners. It is my pleasure to introduce today's speaker, Dr. Catherine McFadden from Harvey Mudd College. Kathy received her BS in biology from Yale University and a PhD in zoology from the University of Washington. Kathy held postdoctoral positions at the University of California, Davis, Port Erin Marine Lab, and University College in Dublin before joining Harvey Mudd College in 2001. She has been a faculty member at Harvey Mudd College since 1991, where she holds the position of Vivian and D. Kenneth Baker Professor of Biology. Kathy's research interests lie in the patterns of distribution and mechanisms that generate biodiversity in the marine environment, with a specific focus on the anthozoan corals, especially octocorals. Kathy and her research group develop and apply molecular and genomic approaches to understand species boundary problems, as well as deep phylogenetic relationships and trait evolution in anthozoans. Kathy is also a keen bird enthusiast. She has traveled and sampled all over the world and is a leading expert in the identification of soft corals. She has previously worked in the Red Sea, but this is her first visit to KAUST. I hope this is the origin of a new, mutually rewarding semi-infinite access. It's a pleasure to welcome you at KAUST, Kathy, and we are very much looking forward to your keynote.
Well, thank you very much, Omar, for that introduction. And also, I'd like to thank Omar and the rest of the Fall Enrichment Program people for inviting me. And it's been a wonderful visit so far. Also, thanks to Mike Berryman and the Red Sea Research Center as well. As Omar said, this is my first visit to KAUST, and it's been lived up to all of my expectations so far. I've been hearing about it for years, but I'm really looking forward to seeing the reefs, which I haven't yet been able to get out to. You all are very fortunate to be living on the shores of a very beautiful sea, and as Omar mentioned, I have worked in the Red Sea, in the Gulf of Aqaba, but just not down here. And I hope that all of you have had opportunities, hopefully multiple opportunities, to get out and snorkel or dive on the reefs and see all the really amazing organisms that are living there firsthand. Now, coral reefs are often referred to as the rainforests of the sea in the respect that they host more species associated with them than any other marine environment. And what I'd like to do today is to talk a little bit about species diversity on reefs and actually how little we know about that diversity, particularly with respect to corals. So I'm going to spend a little bit of time telling you about corals, what are corals, and I think corals are much more diverse than most people realize. We'll talk a little bit about how coral reefs are formed and why they're so important. And then I'll pick up this topic of how many species actually live on coral reefs, and that will necessitate talking a little bit about what are species and how do we go about defining them or deciding that two things are different species, and what are the problems we face in doing that. And finally, I'll finish up at the end with a little bit about why does it even matter? Why should we be concerned with determining what species are out there or how many species are out there? Okay. So corals are very simple animals, and I want to stress that they're animals, right? They often live in tight associations with plants, but the coral itself is an animal, and they are one of the earliest multicellular animals that evolved. Corals in various forms go back probably about 600 million years. And the basic unit of a coral, the animal itself, is what we call a polyp, which you're looking at here, several polyps. And a polyp is really nothing more than a mouth. So polyps consist of a mouth surrounded by a ring of tentacles, and the tentacles have stinging cells on them that capture particles from the water and transmit those to the mouth. And then the rest of the polyp is really just a digestive cavity. So they take in food, they digest it, and any waste goes back out through the mouth. There's just one all-purpose opening to a polyp. So polyps are very simple. They have various different cell types. So there's these stinging cells, digestive cells, reproductive cells, neural cells, but they don't have any organs. So a polyp has no brain. Polyp has no internal organs at all. So in that respect, they're extremely simple. Some corals consist of just one polyp, but the majority consist of multiple polyps that are all connected together. So think of an animal with many mouths. So here's an example of a typical coral. All of these polyps are connected together by tissue and canals at their base. So the food caught by one polyp can be shared with the other polyps in the colony. So this is what we refer to as a colony, an individual with many mouths. And most of the corals that most people are most familiar with or think of when they hear the term coral are the reef-building corals, such as this Aqaba from the Red Sea. And in this case, the polyps are the little pimples that you see on these branches here. So they're arranged around a skeleton. So corals are very diverse. All corals consist of polyps. The polyps are quite similar from one type of coral to another. But what really differs among different types of corals is the skeleton. Whether they make a skeleton at all, what that skeleton is made out of, and the form that skeleton takes. So I want to show you a little bit of the diversity of corals. This is a figure put together a couple years ago showing all the different types of corals that there are out there. And one caveat I want to say is that the numbers of species here are very rough estimates, and hopefully that'll become clearer why as the talk goes on. So we really don't know for sure. These are the numbers of species that have been named, but many species have not been named, and many species have been named multiple times. So these are rough estimates. So the pieces of pie that are shown in red, the two different shades of red there, those are what we refer to as the stony corals or scleractinian corals. So those are the ones that have a hard calcium carbonate skeleton, but only about half of them are actually build reefs. So that's the brighter red color, the hermatypic scleractinians, about 750 species of reef-building corals. That's less than 15% of all corals that build reefs. The majority of corals are the octocorals. Those are shown in blue. That's the group I work on. And they include the soft corals and the sea fans that I'll show you in a minute. And then there's two smaller groups of corals, the hydrocorals, which you may have had the misfortune of meeting. Fire coral, if you've snorkeled on a reef, that's an example of a hydrocoral. And the antipotherians are the black corals. or wire corals is another name for them. So we'll start with the stony corals, and in particular the reef-building stony corals. So here's a typical stony coral in Aqaba. Again, that skeleton made out of calcium carbonate. And again, they take on many forms, such as this brain coral, and these very beautiful complex forms. And here you can see all the polyps that are arranged on that hard skeleton. Here's a fire coral, also a very hard calcium carbonate skeleton. In these, the polyps are very small, very difficult to see. And here's another hydrocoral related to those fire corals. The soft corals, many of the soft corals produce no skeleton at all. They're just large, fleshy masses, sometimes a little slimy. And this is an example. They're referred to as octocorals because the polyps always have 8 tentacles, you can see here. So anytime you see a coral with 8 tentacles, it's one of mine. It's an octocoral. So these can reach very large sizes, often meters across, these big, fleshy colonies that often are very abundant and dominant on coral reefs. The soft corals are also often very colorful, and any photo that you see of the Indo-Pacific often includes a dendronepthia like this one. Again, more complex forms of these soft corals, or these very small colonies as well. The other type of octocoral are the sea fans or gorgonians, and these ones do produce a skeleton, but the skeleton is made largely of protein with some calcium carbonate embedded in it. And in these, the polyps are arranged around these branches formed by the skeleton, but they take on these very complex forms, these networks and fans and large bushy colonies. Again, they can be very large and very beautiful. The final type of octocoral are what are referred to as sea pens, and they're called this because they look like an old-fashioned feather quill pen. These are primarily found on soft sediments such as sand. They have a stalk that they use to anchor themselves in the sand, and then the polyps are arranged on what we refer to as leaves that are distributed around that stalk. Again, these can be very, very beautiful and quite large. And then the final type of coral are the black corals. They also have a skeleton, but in these the skeleton is entirely made of protein. It's this black protein, and it's used in the jewelry trade. So this protein is similar to horn, and it can be polished up to make very nice jewelry. So all of the corals I've shown you so far, again, a wide diversity, occur on shallow tropical coral reefs. But corals also occur throughout the world in other types of environments. So there's many cold water corals, and I've actually worked quite extensively on cold water corals, such as these. Again, they're often very colorful, very large, obvious members of the benthic community. And corals also occur in the deep sea, so down to as deep as 4,000 meters. And these are examples of some deep sea corals. This is from about 1,000 meters in the North Atlantic. Again, these large tree-like corals, gorgonian corals, several different species in this picture. And some of these take on really very beautiful forms. So corals, very diverse. They're found all over the world in all temperatures and depths of water. They're not just on tropical reefs. Okay, so back to the scleractinians. So as I said, corals make different types of skeletons, and the stony corals, in particular the reef-building stony corals, what makes them so important is that skeleton that they produce. So the polyps actually precipitate calcium carbonate, so from calcium ions and carbonate that's in the seawater, through biochemical reaction, they can produce solid calcium carbonate and produce a skeleton that, unlike ours, is not internal to their tissues. It's actually underneath the animal. So they're laying out a skeleton below their tissue. And if you look at this skeleton with the tissue removed, it looks like this. Each of those little pores would support a polyp. And these skeletons are very intricate. The calcium carbonate is laid down in a very specific ways, these intricate structures that help support the polyp. And much of the taxonomy of stony corals and determining what's a species is based on this the details of this calcium carbonate structure. So as the coral grows, it's continually depositing calcium carbonate underneath the tissue, and this gets thicker and thicker over time. Corals can live for centuries, so you end up with these very large, massive structures, such as this huge Porites colony, and there's a scuba diver over there on the left for scale. So this is one colony, probably centuries old, and only the tissue on the outside of it is living, is the polyps. The rest of that is just a huge amount of calcium carbonate. And when the tissue eventually does die, you're left with a solid structure that other organisms can then colonize and live on. And that's really how a reef develops. So we start with corals laying down calcium carbonate, they die, others settle on top of them, lay down more calcium carbonate, and so on, such as you see here on the left, where this kind of coral in the center, tissue has died and has been colonized by other corals that are now living on top of that. Okay. So coral reefs can reach very, very large size over this accumulation of calcium carbonate over the years. Another misconception that I want to dispel here is that coral reefs only occur in shallow tropical waters. Coral reefs are also found in the deep sea. And I had the fortune this summer to be able to see one of these firsthand, going down in a submersible. This is at 700 meters depth off the coast of the eastern United States. And we actually discovered on this dive a coral reef built of this deep sea coral called Lophelia. This reef was several hundred meters tall and estimated to be 85 miles in length. So this is a huge structure that we didn't even know was there until August. And there are more reefs like this we're finding more and more in the deep sea. So corals don't just occur in shallow tropical waters. They're down there, and very important structures in the deep sea as well. Okay. Okay, but for the rest of this talk, we'll focus on the shallow tropical reefs that you have in your backyard. Again, why are they so important? So in the absence of the corals, what you would have out there essentially would be probably a fairly two-dimensional sand or mud habitat. The corals are what we refer to as ecosystems engineers. They generate the entire structure of the reef. Much as in a rainforest, the trees are what makes the rainforest. If you cut down the trees, you no longer have a rainforest, and all the other animals associated with it disappear, die. Same is true for corals. Without them, there would be no solid structure out there, or very little solid structure, and all of the other animals that depend on that three-dimensional structure, highly complex structure, all the nooks and crannies and surfaces of that reef would not be there. So, as I said, many species live associated with coral reefs, either living in holes in the reef, living deep within the reef, swimming on top of the reef, feeding on the reef, and so on. And in addition to all of the various fish and invertebrates that depend on reefs, humans also depend very heavily on coral reefs. These figures are a few years old, but it's been estimated that about 275 million people in over 100 countries depend directly on coral reefs for their sustenance and livelihood. And that's particularly true around the equator and in many developing parts of the world. Strong dependence on coral reefs. The economy of reefs has been quantified, $375 billion a year economy associated with coral reefs, millions of jobs dependent on them. Those are in areas from commercial fisheries, recreational fisheries, tourism in general is a huge economic generator of economy associated with reefs. And then simply subsistence fisheries in many parts of the world where people are directly dependent on the reefs for their own food and livelihood. So given that there are many species that are dependent on reefs, said we know they're extremely diverse, how many species actually live on coral reefs? Okay. And the answer is simply that we really don't have a very good idea. There have been some recent studies that have attempted to quantify through various different means how many species live on coral reefs. And those estimates range from 500,000 to 1 to 3 million, 1.5 to a bit over 3 million, and anywhere from about 500,000 to 10 million species that might be living on coral reefs. So those estimates range over an order of magnitude and show you how little we yet really know about how many species are living on reefs. How many coral reef species have actually been described? And by described, I mean there's a scientific description, an official name has been given to a species. Again, it's a little difficult to quantify accurately, but it's been estimated somewhere around 95,000. Okay, and if you use those sort of 1 to 3 million estimates as what might actually be out there, that's only about 9% of what's actually living on reefs has actually been formally described and named and is known to science. Okay? Which means that over 90% of what's out there remains undescribed. And that holds for the marine environment in general, not just coral reefs. So why is that? Why do we know so little about what's living on reefs? We've been studying reefs for quite a long time. And I think some of our other speakers have addressed or will address some of the major factors contributing to how little we know about reefs. Luis on Sunday talked about these habitats that are simply very difficult to access. Deep sea is one. These mesophotic twilight zones he talked about are another. It's just very difficult for humans to get there and work there. We don't survive underwater very well. And those areas simply remain unexplored. A second problem that I think this evening's speaker, Gustav Pauli, will talk about is that many organisms are simply difficult to collect. So much of what's out there, probably the majority of all this undescribed biodiversity, are very, very little things, tiny crustaceans and worms and things that are living deep within the reef, and we simply don't see them and can't collect them very easily. Another contributing factor is we simply don't have the manpower to describe what's out there. There are too few people, too few taxonomic experts in many of these groups. Just simply very few people who are working on many of these organisms. The group that I work on, these octocorals, they're big, they're colorful, they're charismatic. The number of people in the world working on them is about the number of fingers on one hand. Okay? And then the question that I really want to address in the rest of the talk is the issue of cryptic biodiversity. And that's simply that many species have not been recognized as being different from others. So even among those 95,000 or so that we have named, and some of which we consider very well known, very well studied, we're learning more and more. We actually don't know very much about them, enough to know whether they really are different species or not. And we're finding that our ideas of what species are have been changing greatly over recently. Before I talk about this cryptic biodiversity, I want to just take a second to talk about the term cryptic. As cryptic is a word that is cryptic. It's confusing, and it is used in different ways even within the field of biology. And there's at least three different ways that the term cryptic is applied to the organisms that we find on coral reefs. And I'm going to be specifically talking about one of those ways. So one way in which cryptic is used, cryptic really means hidden. So there's many ways that organisms can be hidden from us. So cryptic can mean camouflaged. Okay? So this is, what you're looking at here is a snail that is living on a coral. And that snail is very camouflaged against the coral. It actually mimics the coral that it is living on to the degree that it has fake polyps on it. And you know they're fake if you count the tentacles because it didn't quite get the number right. But that snail we would refer to as being cryptic. It's camouflaged and hidden against the background. Okay, so that's one use of the word cryptic. Another use is hidden in the sense that things are out of sight, in this case, deep in the reef. So again, I think Gustav this afternoon will introduce us to the cryptobiota of the reef, these little organisms that are living deep inside the skeleton of the reef, and we simply don't see unless we break the skeleton apart. And the third use of the term cryptic, which is the one that I'm going to be discussing with respect to corals, is one that really means lookalikes. And again, Omar mentioned that I have a strong interest in birds, and birds are very good examples of what we refer to as cryptic species. So by cryptic species, what we mean are things that look so similar to us that we've thought they were one species, but it turns out that they're actually not. And we've been learning more about that through a variety of means over the last few decades. So this is a bird that's very common in North America and also throughout northern Eurasia. It's common, it's been known for hundreds of years, and until 10 years ago, it had been considered to be a single species found throughout that whole global area. And only recently it's been realized that there are actually not even two, but three species. So the one that's in Eurasia, it's a different species. In North America, we have two. The winter wren that occurs on the east coast of North America, the Pacific wren on the west coast. Now, to us, I think they look very similar. But they're different species, and this is what we would refer to as a cryptic species, things that look alike, at least to us. Okay. So before I talk about cryptic species in corals and other marine organisms, again, we've got cryptic species in fish and all of the other invertebrates that are out there on the reef. Before I talk more about them, I wanted to talk a little bit about species, how we actually define species, what is a species, and how we go about determining whether two things are different species or not, and why that can be a kind of a difficult problem. So, the definition of species that most biologists, I think, adhere to or strive to adhere to is that of reproductive isolation. And by that, what we mean is that if two individuals are members of the same species, they could meet and mate and have offspring together, so they can reproduce. If they're different species, they may never meet. They may meet but not be able to mate. They may try to mate but be unsuccessful in producing offspring. So they would be different species. They'd be reproductively isolated from one another, unable to reproduce together. And there's slippery complications with this definition in some species, but for the most part, that's sort of the ideal definition of species that we as biologists have kind of strive to adhere to, groups that are reproductively isolated from one another. So how do we determine that? How do we decide if two individuals are reproductively isolated or not? So one way, the most direct way, is just to see what's going on out there and see who's mating with whom and whether they're successful at producing offspring. And this can be done in some organisms, like, again, many birds. You can go out and see who's paired with who and whether they're successful at raising young. But in the marine environment, this is very difficult to do. Many organisms, including most corals, when they reproduce, it's not a matter of a male and a female getting together and going off in a corner somewhere. Instead, what happens is all of the individuals, perhaps on a reef, will release their eggs and their sperm into the water in response to particular cues, and fertilization simply occurs by a sperm finding an egg in the water. And many species will reproduce at the same time, so you have this whole mix of eggs and sperm all mixing around in the water, and it's essentially impossible to know what sperm got together with what egg and where they came from originally. So actually determining who's reproducing with who is very difficult in these types of organisms. So the second and really the most common time-worn method of determining whether organisms are reproductively isolated or not is to look at their morphology, which simply means their shape or their form, their physical appearance. And this is the way species have been described for the last several centuries, is if two things look sufficiently different, we consider them to be different species, assume that they're reproductively isolated. If they look very similar, we consider them to be the same, and again, assume that they are not reproductively isolated. But there's difficulties with this. I've already shown you the cryptic species problem. Things can look very similar, but nonetheless be reproductively isolated, be different species. We have the flip side of that as well, which is that within species, we know that there is variation among individuals, and one very common species illustrates that very well. So there's a lot of morphological variation within our population, within our species. We know from direct observation that any one of these individuals could reproduce successfully with any other, regardless of the variation that we can see here. But we don't necessarily know that for other species, where we see differences like this from one individual to another. Does that mean they're different species or not? So both of these things confound our use of simply the physical appearance of organisms to determine whether they're different species. Things are either too similar or there's a lot of differences within a species, despite the fact that they can still reproduce together. So the third way we've been assessing reproductive isolation, really just for the past, I'd say 3 decades or so, is to use genetic information. And we're getting better and better at this as technologies develop further and further. So we can now take 2 individuals and look at their DNA, either the DNA for a particular gene, or now we can look at the entire genome quite easily, or some large fraction of the genome, and see how much of a genetic difference is there between individuals. We run into some of the same problems that we do using morphological differences, which is, okay, well, how different do 2 individuals have to be genetically to still be the same, to be different species, or how different are individuals of the same species genetically. But it's been really the advent of this methodology, being able to look at the underlying genome of the organisms, that has led us to recognize how flawed our interpretation of species based on morphology has been. So I'm going to give you a few examples, both from stony corals, work that's not my own, and then a little bit from my own work on the soft corals, in which looking at the genetics has really illuminated us and caused us to realize that our assumptions about what different species were based on what they look like are often deeply flawed. And I'm going to start by quoting our speaker from 2 days ago, Luis Rocha, who works on fish, and he said, I think he thought this was going to be like a slur, but corals are a total mess, and I don't know anybody who works on corals who would disagree with this statement at all. So I'll show you why corals are a total mess. Okay, so as we said, one of the ways that species are confused with one another is if we have morphological variation within a species, but we don't recognize that and think that those morphological differences indicate things are different species. So here's an example from work from Rob Toonan's lab in Hawaii, published a few years ago, in which they worked on a common reef-building coral that occurs in Hawaii, the genus Montipora. There's a number of species of Montipora that occur there. Some of them are very common and widespread, and some of them are considered to be very rare. So shown here are 3, what had been described anyway, as 3 different species, Montipora flabellata, turgescens, and dilatata. And from the photos of the growth form, you can see they look kind of different, right? They have different growth morphologies. And dilatata was of particular interest here because it's very rare. It's only known from Hawaii, and I think from just like one bay in Hawaii. And the US government was considering it for listing as an endangered species, which would provide it with all kinds of legal protections. So Toonan's labs sequenced these different species and looked at the genetics and discovered that genetically they're all the same. Okay, so what had been described as 3 species on the basis of the fact that they look quite different are really all the same species, probably growing differently in somewhat different environments. And what was thought to be this very rare endangered species is just an unusual growth form of a much more common, widespread species. Another very interesting study that showed that not only do you find variation among individuals of different corals that can confuse you about whether they're different species or not, but even a single individual can confuse you. So this is a really interesting paper published a few years ago by David Paz in Mexico. And he worked on Posilopora damicornis, which is an extremely common reef-building coral supposedly found throughout the Indo-Pacific. And there are a number of other species of Posilopora that also occur in Mexico, including Posilopora inflata, which again is something that was considered rare, only known from Mexico and Central America, probably a sort of special species endemic to that area. So what David did was he was photographically monitoring colonies of Posilopora damicornis, going out monthly or some periodicity and photographing the same colonies over and over again. And you see one of these colonies at 5 different time points in these photos. So for the first 2 years, these colonies maintain the typical form of Posilopora damicornis, which has these quite fine branches, fingers on it. And then sometime in 2013, there was an environmental event, actually a big storm that deposited a very large amount of sediment into the bay where these corals were living and impacted the reef there. And the corals he was photographing turned into Poeciloptera inflata. So they went from having these skinny little branches to having these big, fat, swollen branches that are a characteristic feature of Poeciloptera inflata. And then a year later, they went back to being the typical Poeciloptera damacortis form. So here, not only is this all one species, but one individual changed over time to a different species and back. So again, inflata is not a different species. It's simply a growth form of Poeciloptera damacornis that it adopts apparently in response to the environment. So those are some examples where, again, we've thought things look different, described them as different species, and then subsequently learned that they're actually all variations of the same species. Turning now to the opposite, to the cryptic species issue, cases where what we thought was one species, widespread, we've now learned maybe multiple species. And again, an example from reef-building coral, Stylophora pistillata, very common reef-building coral, especially here in the Red Sea. And it's known distribution from the Red Sea, southern Indian Ocean, all the way across to the western Pacific. And there's some morphological variation if you look across that range, again, in the thickness of the branches and how knobby they are and things like that. So Ellen Chen's lab from Taiwan, along with a huge host of collaborators from across the Indo-Pacific, did a detailed study of this species a couple years ago. And they looked both at the details of the skeleton and also, again, at the genetics of Stylophora from these different regions and discovered some very distinct genetic groups. So in this case, the Stylophora from the western Pacific, but the top there, and from the kind of central Indian Ocean were two distinct groups, kind of related to one another, but more closely related genetically to a different species, actually a different genus, Seriatopora, than to the other groups of Stylophora from the southwestern Indian Ocean and the Red Sea. And it's looking like this could be, again, a cryptic species complex of potentially four different species that are not widespread throughout the Indo-Pacific, but each one localized to a particular area. So we find one group in the western Pacific and eastern Indian Ocean, and then three of these different groups here in the western Indian Ocean. What's here in the Red Sea and also in the Gulf of Arabia is something genetically quite different from what's in the western Pacific. Okay, so those are some examples with the reef-building corals, and I'll show you a couple of examples from my own research with soft corals, particularly here in the Red Sea. So this is up at the Gulf of Aqaba, shallow fringing reef, and what you're seeing in this photo is probably 2/3 soft corals and 1/3 stony corals. And most, if not all, of the soft corals in this photo belong to the genus Sinularia, which is the Acropora of the soft corals. There's about 170 described species of Sinularia. Any one reef in the Indo-Pacific, we typically find anywhere from 10 to 35 species of Sinularia living together on the same reef. They're very confusing and very difficult to tell apart from one another. And they've been worked on here in the Red Sea for a long time. Many corals, both soft and hard corals, were first described here in the Red Sea. People have been working in the Red Sea longer than anywhere else on tropical reefs. So we looked at a couple of these kind of common species of Sinularia from the Gulf of Aqaba. And one that you find up there is the Sinularia compressa, which was believed to be found only in the Red Sea, so endemic to the Red Sea. And then Sinularia polydactyla is a species that is considered to be common, widespread, occurring throughout the Indo-Pacific. But in the Gulf of Aqaba, we found two different forms of Sinularia polydactyla, the one in the top right there, which has these short, knobby kind of fingers, and the one in the lower left, which has these much longer, less dense fingers, and based on the other kind of skeletal characters that are used to distinguish species, these both fit the definition of Sinularia polydactyla. If you go out to the Western Pacific, to Palau, you find, again, things that also fit the definition of Sinularia polydactyla. They also have these long fingers, even more slender than the one in the Red Sea. So we did some genetic work to look at these different forms, and what we found was a mess. So one thing we did, so Sinularia polydactyla was described from the Red Sea in 1834, and we went back to the original museum material of what was originally described at that time and compared it to these our modern living species, and discovered that actually Sinularia compressa is Sinularia polydactyla. So what we had been calling Sinularia compressa actually is the original Sinularia polydactyla that was described in 1834. So the name compressa is redundant. Get rid of that and recognize that now as what is true polydactyla. The other things that have been being called polydactyla are different species. So we've now described a new species from the Red Sea at the top there, a Sinularia levi, and the second one down, with one of the long fingers, is undescribed. We haven't described that one yet. It's probably a new species for the Red Sea, but it's not Sinularia polydactyla. And then what's been being called Sinularia polydactyla in the Western Pacific is probably at least 2, if not 3 different things, and we're still trying to sort out whether those are, again, new undescribed species or if they're just things that have been misidentified. So there's names that are available to be applied to them, but we just don't know what those are. So what we started out with thinking there were these 2 species, compressa and polydactyla. Turns out compressa was a misidentification of polydactyla, and the other polydactylas are, again, a group of cryptic species, probably at least 5 distinct species, but 3 of them, well, polydactyla plus 2 of these others, found here in the Red Sea, and probably only in the Red Sea. Okay, my final example is a group of soft corals called the Zinnias, and this is a family of about 140 species of very small, soft corals. But despite their small size, they often form large carpets over the reef. And I've just been told that there's areas that have bleached out here that are currently overgrown with carpets of these Zinnias. Zinnias are of increasing interest. I think for this reason, it's been found that they are very good at colonizing reefs that have been disturbed, either through bleaching or other types of events. They recruit very quickly, cover large areas of surface, and it's been speculated, but not yet really shown conclusively, that they are likely to be inhibiting the recruitment of stony corals back to these disturbed areas. They are also, now we're learning, invasive. They're popular in the aquarium trade, and we have at least 3 instances now where Zinnias from the Indo-Pacific have been introduced into the Atlantic, where they don't occur, and are now completely overgrowing coral reefs in Venezuela and Brazil. And if you want to see some a really sort of startling example of invasive species, go on YouTube and Google Zinnias in Venezuela, and you'll see a video that goes on for many minutes of someone diving over a reef in a national park in Venezuela where Zinnias have covered everything, including all the fire corals, all the scleractinians, is nothing but Zinnias. So we have some interest in this group. And the problem with them is it's very difficult to distinguish species from one another, and we've resorted to doing this simply genetically. Now, again, Zinnias have been worked on in the Red Sea longer than anywhere else, and we know a lot about them in the Red Sea, and we can put names on the ones that are here in the Red Sea, but we can't put names very easily on others. And the names, some of the species that have been described from the Red Sea, those same names have been applied all the way across the Indo-Pacific. Again, the assumption that what's here in the Red Sea is also the same thing that we're seeing in Indonesia and Australia and so on. So we've just done a large genetic study of Zinnias from across the Indo-Pacific, about 13 different locations at this point. This slide's a little complicated. I don't want to get into all of the details, but essentially here, every every branch tip is a species that we've defined genetically. Every colored dot is a location where we found it. What we've discovered is most things are only occurring at a single location. So we find a species and we've only found it at one place. We're not finding them widely distributed throughout the Indo-Pacific. And this is particularly true of the ones from the Red Sea. So I've highlighted here 4 species that have been known, again, were described several hundred years ago from the Red Sea and have been assumed to occur throughout the Indo-Pacific. Zinnia umbellata, Sympodium peruleum, and Thelia glauca. And now, so the arrows are pointing to those species from here, Kaust and in Israel, the Gulf of Aqaba, where those fall here. Those 3 species are unique to the Red Sea. So the things that have been being called those names elsewhere are genetically very different. They're different species. So those 3 are endemic to the Red Sea, only found here. Sympodium is a particularly interesting example because it had been thought there was only one species of this genus Sympodium, the same species you found everywhere. It looks like from these data there's at least 9. One species occurring in the Red Sea and only in the Red Sea, but at least 8 species occurring elsewhere in the world, none of them the same as what's here in the Red Sea. And we do have one species, that Heteroxenia fuscescens. Again, it's very common here, and we find it everywhere. So for whatever reason, that one is extremely widespread, but the majority of these xenids are very localized in their occurrence and are only occurring in particular locations. And again, the ones here are unique to the Red Sea. Okay, so I just want to finish up with a little bit about sort of why does this matter. Again, what I hope I've shown you is that corals are a total mess. We know little about the biodiversity of reefs. Again, in a lot of cases, that's because it's simply difficult to access the environment and access the organisms, and we don't have enough manpower to really describe what's out there. But what I've been telling you about today are the things that are well known, that are large, that we can see. We still don't know enough about them to really conclusively know what the different species are. So why does that matter? Whoops. Did I? Okay. Again, others have talked about the crisis facing coral reefs currently. I think it's well known that there's severe environmental impacts currently on coral reefs, and predictions that we may lose a large fraction of the reef-building corals anyway within this century as a result of ongoing climate change and other environmental impacts. And I think we as scientists anyway would like to try to both understand what's going on, predict what might be the future of our reefs, and help to mitigate and conserve and protect our reefs from these changes. But in order to do that, we need to know who the players are. I mean, the reefs are simply a conglomeration of the species that are there, and understanding how a reef behaves in the face of environmental change involves understanding the responses of those individual species to change. So just as an example, here are some soft corals that are bleaching, and this was a reef where most stuff was completely unbleached and healthy, and here was this little pocket of bleaching, and I have no idea whether those 3 individuals that are bleaching are the same species as the one in the right-hand corner or not. But to understand who's surviving, who's doing well, who are likely to be the survivors in the future, and who's doing poorly, we need to know who these species are and be able to distinguish them from one another. And I think in particular our efforts to protect coral reefs and protect the species that live on them really depend on knowing what species occur where. Examples where this can be not knowing what the species are that occur where and whether they're the same as what's occurring elsewhere can be problematic. For example, results of research conducted in one geographic area may not be applicable to other areas if the species are different. Stylophora pistillata, I think, is a very good example of that. A huge amount of research has been done on that species here in the Red Sea, but if it's a different species from what's in the Western Pacific, can we assume that what we understand about that species from the research here is applicable to what's in the Western Pacific? It's not clear that we can. So simply to sort out what's going on, we need to know whether we're working on the same species in different areas or not. Particularly important also, I think, is that endemic species, so species with narrow geographic ranges, have been shown to be more vulnerable to extinction than geographically widespread species. They're often more finely adapted to a particular habitat. They're more vulnerable to a localized environmental event that might wipe out all of the, the reefs in a particular area. And if they're endemic, they're unique to that area, there's not the possibility for those reefs to be recolonized from somewhere else. Okay? It's different stuff elsewhere. And I think that's something particularly to keep in mind here in the Red Sea. The more research we do here, the more we're realizing, I think, how unique the species in the Red Sea are. Again, research has been done here on coral reefs the longest. We've assumed that stuff that was first described from here is the same as what's elsewhere in the Indo-Pacific. That's looking like it's not the case. In fish, in corals, most likely in all the other invertebrates as well. So the reefs here are unique fauna, what happens here in the Red Sea is going to impact things that can't recolonize or be artificially recolonized from elsewhere. It's different stuff elsewhere. So, so I think I just want to conclude by saying that the Red Sea has many more endemic species than previously thought. Okay, we just haven't recognized them as being different from what we see elsewhere in the Indo-Pacific, and they're essentially irreplaceable. So if you lose them here, we're not going to get them back from anywhere else. And I think that's something to bear in mind. Both with the research that goes on here is the applicability to other parts of the world, but also in particular to take a special care of the reefs that you have here, because they are very unique, much more unique than we thought. Okay. And with that, I think I will stop and thank you all. And if we have time for questions, we'll take some questions. Go ahead.
We do have time for questions, but if you'd like to pose a question, wait for the microphone. A little bit like the reefs, we have two kinds, the soft kind, which you may throw, and the hard kind. Please don't throw it.
It's mine. Sorry. Thank you for the excellent presentation. Did you find any endemic species to the Gulf of Aqaba compared to the Red Sea, only in the Gulf of Aqaba?
I haven't done enough work yet in the Red Sea to know that. So we have very good information from the Gulf of Aqaba, the Israelis have been working there for many, many years, and I've been working up there, but it's 14 kilometers of coastline. So we know very well what's there. I have, at this point, very little data from here, just a little bit. What we have so far for the soft corals suggests genetically that what's here is the same or very similar to what's in Aqaba, but really very little surveying has been done here, and I'm really waiting to get out on the reef on Thursday to see what's here and do some collecting and start to really compare what's here in this part of the Red Sea to the Gulf of Aqaba.
Thank you very much for a great talk. There are several corals gives the fluorescent signal in the night. I just wondering if you already analyzed those corals for sequencing data, and I'm just wondering if these coral makes a kind of the colony in the genetic tree or not. And second question is just simply why these corals need to give the fluorescent signal or not. Why is that? Why they need to have this kind of the different color?
Boy, that's way out of my research area. I'm sorry. The fluorescence is not something that I have worked on. It's very interesting, and as you may know, there's information showing that when these corals are very stressed, like when they're about to bleach, they give off these fluorescent signals, or colors change. But I think work is really just sort of starting on that, and I haven't done any work on that. There are some of these soft corals are actually not just fluorescent, but actually bioluminescent, and particularly some of the deep sea ones are bioluminescent. If you the Ranilla luciferase system, which is used in molecular biology, is a bioluminescent protein that comes from a sea pen. And why they're bioluminescent, I have no idea why that would be. And I don't think anybody's really looked at that. On that. Where's the? Go ahead, whoever's Sarah. Yeah. Yeah. Okay. Oh, hi. Go ahead.
Hello? Yes.
Yeah.
Okay, I think you might have been going to comment on this, but what level of genetic differentiation are you using to decide something's a separate species, and why?
Yeah,
and why?
so you may know, so the tree that I showed is just basic DNA barcoding with 3 different markers. As you may know, DNA barcoding has been a real problem with corals because the markers that are typically used for that are very conservative, and it's been very difficult to find markers that we can use to delineate species in corals. So the actual threshold that we're using with this 3-marker system is 0.3%. But it accords quite well with our morphological species definitions. We get a quite high agreement with this 0.3% genetic distance threshold and what we think are different species based on morphology. We've now are in the process of developing a system, a target enrichment system, where we can sequence about 2,000 loci in an individual and then get much more detailed information on species boundaries, but we're still developing that system.
Thank you for your talk. It was very interesting. You've talked about a few species that looked very different but turned out to be the same. Does that mean that their function is also more similar than we thought, or do they still provide different niches or different food or for different species? I mean,
that's a good question. I don't know, and I don't know that's specifically been looked at. I would guess that these stony corals, where the differences you often see are in the actual physical structure, the thickness of the branches and things like that, potentially would support different species living in or on them as a result of that kind of different 3-dimensionality they offer. But I don't know that anybody has actually looked at that particularly. It's a good question.
Hi. With all the development, man-made development in this, in the Red Sea area and with global warming, are we seeing any damage done to the coral reef here? Bleaching or effects?
Again, I haven't worked here. I'm told that there is bleaching going on right now on the reefs out here. I know there's been quite severe bleaching in the southern Red Sea. Again, I don't think that's a function of development. I think that's a function of climate change and warm water events, most likely. But the people who are actually working here on the reefs here can answer that a lot better than I can on that.
So you showed a pretty interesting study that you have different morphology, or you have different forms of the same species, right? And if one of those forms is endangered, but it's all one species, is it worth protecting that form if that's occupying a different sort, or giving a role, like she just asked?
Yeah, that's a really good question, and I think there's, unfortunately, at least in the U.S., our endangered species laws are based on defining species as reproductively isolated groups, but there is certainly an awful lot of value in conserving just genetic diversity, period, whether or not things are different species. If it's a genetically distinct population, then I would say yes, absolutely. That would be my reaction would be yes. We should conserve as much genetic diversity as we can, regardless of whether that genetically diverse population is technically a different species or not. But that's not the way the government works in terms of legally protecting species. On that.
Thank you very much. Let's thank Catherine again, and maybe I ask you to stay on stage here for us to present a trophy commemorating the lecture.
We have to go over here? Okay. Thank you.